Hey guys, I'm Sachin.
And I'm Saurabh.
We are Team GeNLearn. Welcome to our experimental demonstration.
Our vision is to enable personalized medicine that is affordable and could be made widely available. The DNA of an organism contains all the information required for its creation, sustenance, and development. Genomics is the study of gene expression, the natural process occurring in all life forms, in which the information in the DNA, that is the genotype, is translated into the physical traits of that organism, also known as phenotype. This could lead to a plethora of possibilities in the domain of personalized medicine, as the person's phenotype potentially contains information about the various aspects of the person's health, for example, allergies, predisposition to several diseases, and compatibility with several medications. Gene networks are required to be used to perform inference on a per person basis based on this information. Bayesian networks, which are a type of probabilistic graphical models, are excellent formalisms to model the gene networks and enable asking questions such as those in a personalized medicine context. The process of learning the Bayesian network, which is the most accurate representation of the gene network from the gene expression data though, turns out to be challenging and of high computational complexity. A fundamental question we ask here is, can we perform the discovery and inference at a performance level that allows us for a widespread low-cost usage model? Can we personalize cancer diagnosis and treatment, which can be made widely available in hospitals? This is currently not possible due to the complexity of the gene networks as expressed for Bayesian. We believe that this can, however, be enabled with a solution where critical parts of this approach are significantly accelerated with hardware.
In this project, we use the D10 Nano SoC FPGA development platform to design and demonstrate an embedded system that is specialized to perform Bayesian structure learning for gene network targeting breast cancer. The approach can also be extended to other diseases and medical applications for humans and other life forms. In our design, the software program running on the hard processor system HPS searches over the space of candidate Bayesian graphs. These candidate Bayesian graphs are then mapped onto the logic blocks in the FPGA for a complex graph-based scoring task, after which a single score for each structure is returned. The software program keeps track of the scores for each Bayesian structure returned by the FPGA, and ultimately the structure with the highest score is chosen as it is the best representation of the underlying data. Since the sampling-based scoring operation can be highly parallelized in the FPGA, and we can perform soft reconfiguration between the candidate structures, an exponential speedup is achieved when compared to software-only implementations. We demonstrate structure learning of a breast cancer gene network with the expression profiles obtained from the gene expression omnibus dataset using the proposed system. We successfully validate the network and extract its performance benefits versus our software implementation in our programming language on an 8-core 16-thread Xeon workstation. We show that our implementation achieves 5 times speedup as compared to the software implementation. Let us now move on to the experimental demonstration. Now we'll go through the experimental setup. As you can see, the D10 Nano board is all powered up and connected to the PC through the USB serial communication. I have also turned on the console of the operating system on the board. I have cross-compiled the program and it is ready for execution. I'll go ahead and execute the program. As you can see, the program read in the patient expression data and it is starting the execution. In some time, it will print the final structure obtained by the program on the console. This is the final structure obtained after the execution of the program as seen on the terminal. The same structure is seen here in its graphical form on the right. On the left, you can see the reference structure from the KEGG gene pathway database. The final structure generated by the algorithm is compared with a reference structure from the KEGG gene pathway database to validate the approach. The KEGG database is an extensive library which maintains and updates a large collection of reference gene networks. Note that such comparison is not expected to be available for all applications, but we chose this specific gene network such that we can prove our design. As evident from the gene network graphs, the GeNLearn implementation does a good job in discovering the gene network. This implementation is not an exact copy of the KEGG reference gene network, as the reference gene network is formed by compiling the results of several research efforts. Furthermore, for this implementation, we considered a relatively small database with only 3,000 patients. This is, however, is not a limitation, as the qualitative benefits when scored across a larger dataset would persist.
In conclusion, we have demonstrated accelerating the Bayesian network structure discovery of breast cancer gene networks using a gene expression dataset of 3,000 patients. When multiple instances of candidate structures are scored in parallel, our system already indicates a 5 times performance improvement versus software implementation. Furthermore, a larger gene network would show an increased performance benefit due to the exponential improvement as size increases. Our prototype platform demonstrates the feasibility of realizing a relatively low-cost system for targeting key applications in personalized medicine. Our ultimate vision is a personalized medicine on a stick. Such system would be able to perform low-cost and high-performance Bayesian inferences toward personalized diagnosis and treatment of cancers. Thanks.